ब्लड नाही एवढं काय डोक्यात घे नाही का इकडे जाईल ना
That's okay.
Hello. Hello, Avinash sir.
Yeah, can hear you.
Sir, can we start?
Yes. As and whenever you want, I'm ready.
Okay, thank you. I request all the participants to switch off their microphones and switch off their video mode. And the participants who are unable to join us on Google Meet can join us on our YouTube channel, which is Webinar DUBSSC, and the link is shared in the chat box. Very Good morning, everyone. In Dapoli Urban Bank Senior Science College, I welcome you all in the national webinar on the significance of the ecology and biology in oil and gas projects, which is organized by the Department of Environment Science of Dapoli Urban Bank Senior Science College, Dapoli. We graciously welcome our national as well as international participants for this webinar. Which makes us feel that people are very much concerned about today's environment. The webinar will begin with the inaugural speech of our principal, Dr. S.P. Jagdale sir, and followed by the convener, Mrs. Amruta Mohite, HOD of Environmental Science Department. Now I request Dr. S.P. Jagdale sir to start his inaugural speech.
Good morning. I'm Principal Jagdale of Dapoli Urban Bank Senior Science College. I welcome today's guest, Avinash Lip, who is an international scientist who has contributed much about Nigerian ecology. I welcome Environmental Science Department and IQAC of the college and dear participants. Today's topic is very relevant, the significance of ecology, biology in oil and gas projects in Ishuran Ecology, Nigeria, and he will be referring also the Indian Oceans. This particular topic is very demanding, and our Department of Environmental Science has organized this particular webinar. Let me introduce the college who is the organizer. This college is located in Dapoli, District Ratnagiri of Maharashtra State. College is now teaching 6 subjects in undergraduate level and 5 subjects at the post-graduation level. About 250 students are enrolled for PhD. Department of Botany and Zoology is recognized for PhD studies. So we are. This is our almost 24th webinar of the this college, and we are organizing on different topics. So in future, we may approach for different webinars with you. Kindly participate. We'll try to make it international. Today, I welcome behalf of Dapoli Education Society and Dapoli Urban Bank Senior Science College, Department of Environmental Science and IQAC of the college, to our chief speaker today, Avinash Dulip. So I will not take much time of you. I request to start the webinar. Best wishes from my side. Thank you very much.
Thank you so much, sir. Now I request Mrs. Amruta Mohite ma'am, who is HOD of Environmental Science Department, to speak few words.
Good morning, everyone. Good morning to one and all. I welcome Dr. Avinash Tulip, sir. It's been an honor to have you, sir, for this webinar session. I also welcome all the participants and the staff members of Dapoli Urban Senior Science College. First of all, I would love to thank the principal of the college, Dr. S.P. Jagdale, sir, for giving us this opportunity of conducting a national webinar. I would also thank Vice Principal of the college, Dr. Sathe sir, IQSC Coordinator, Dr. Ghalme sir, the Technical Department, Mr. Dongre sir, and his team for all the technical support during the webinar session. Today, we have gathered here on this online platform because of the environment which has got disturbed, and hence it needs some time to recover itself. That is what we are going to give it during the lockdown period, is some time to recreate itself. So please take care of yourself and the people around you and let the nature do its work. These days, students tend to search for different new courses This degree in Masters in Environmental Science will be the right platform to take part in, where students from different streams like microbiology, agriculture science, biotech, chemistry, zoology, botany can apply for this master's degree. This is how, once again, I welcome you all for this webinar session, and I'm eagerly looking forward for it to start. Thank you, sir.
Thank you so much, ma'am. Now I invite eminent speaker for today's session, Mr. Avinash Dulip sir. Mr. Avinash sir is freelance advisor and consultant for environment and safety petroleum industry. He is an ecologist, knowledge broker, mentor, innovation catalyst and entrepreneurship strategist. He has 35 years of experience as safety and environment specialist, work in Colombia, Brazil, India, Australia and many other countries. He has worked as petroleum regulator with National Petroleum Authorities. He has provided guidance on Equator Principle for managing social and environment risk. He has worked with public sector companies like ONGC, Reliance, World Bank, and many more. He is also an author for a book called Be Someone Thinking with You, which is a book on self-assessment. This is only the small portion of his achievements, and the list remains never ending. It is our honor to have Mr. Avinash Dulip sir as our resource person. Now I request Mr. Avinash Dulip sir to start the session and share his screen.
Yeah, first of all, I'd like to thank the Dapoli Education Society Science College, particularly the principal, Dr. Sandesh Jagdale, Mrs. Amruta Mohite, who is head of Environment Department, the organizers, to have given me this privilege to give this talk. I also like to thank all the participants. From the chat box, I realized that I have a lot of people from different backgrounds. I have students who have been studying in zoology, botany, environment sciences. I also find out from the chat box that we have professionals who work as environment officers in various companies. And surprisingly, I see a lot of list of eminent professors, senior lecturers, faculty members in various colleges and institutions, particularly in India and a few from abroad. This topic, I'm trying to see. Give me a minute. I'll just try to put on the full screen. Oops. Are you able to see the screen now? Just trying to find out. Yes. Are you able to see the screen? Hello?
No, sir.
Can you hear me?
No, we couldn't.
Okay. I'm trying to get my screen on.
Sir, you can share your screen.
Yeah, I'm trying to see where to share it from. I'm sorry.
Okay, you can go on. The option is on the right side, which is present now.
Yes.
You can click on that and you can select the option which is your entire screen.
Okay.
Okay, and just click on the window and share.
Okay. Now it's there.
No, sir.
Okay. One sec. I'll just get there. Share. Okay.
Yeah, sir.
Sorry, audience, for a small break. I was just trying to organize a few things on my screen. So I have to thank all the participants, like I said earlier, and welcome you to this very interesting topic. When the principal, Dr. Jagdale, asked me to look for a topic to discuss, I was thinking, I'm going to work in 35 years in the oil industry. I would prefer to talk of something more important because there's a lot of information already on the oil industry and how it manages the environment. So I chose this topic, the significance of ecology and biology in oil and gas projects. The reason I chose this is because you need to understand that this is a subject that we seem to be very much aware of, but very little we seem to understand. And looking at this, we look at biology to mean a study of all life or living materials, and ecology to mean the study of all life and the interaction with the environment and among themselves. Makes it very interesting for us to look at. The topics that I'll be covering today are basically two. I will start with a few things on environment concepts, basically related to ecology. I feel it's something that we take for granted, and we need to understand them a little bit much better with reference to how a development project like oil and gas works with it. I'll be covering estuarine ecology in the Niger Delta. I'll be sharing with you actually my master's degree thesis on estuarine ecology, basically biogeochemistry of estuarine, environment where I studied. It is still relevant to what we see today in estuaries. Estuaries are very important ecosystems and very complex. So I'll be covering a few points on that. And then subsequently, we will go to the marine ecosystem, in particularly the Arabian Ocean. And here I will see with the vast amount of oil and gas activities in the last 50 years or 40 years offshore, how we are able to monitor the environment in which we are operating. I start with the basic question, why do we want to think about biology or ecology? For a lot of students studying life sciences, it may have been an interest, a genuine interest to understand biology or zoology or environment. It could also be a simple reason to be a graduate. But I'm going to be challenging your minds because this is not a subject that only ends in concepts. It's a subject that actually intricates into the industry, intricates into development, intricates into economy. So we will look at it. I will also use this privilege to challenge the minds of the consultants who actually prepare environmental impact assessments, the companies. They have reached a certain level of expertise where numbers and figures and modelings make more sense for them to look at impact assessment. But I want them to rethink and look at biological systems more from the point of view of the project setup and see how they are related to the project. I also have an opportunity to challenge the regulator on how you look at biological systems while monitoring a project. Two things came to my mind when I was looking at this particular topic. As a regulator working in a certain country, I used to read a lot of environment impact assessment reports from operators, environment management reports from operators, and I realized that a lot of information and data is in these reports, but there is an absence of information on the actual ecology or biology where the setup or these facilities are located. I went asking the operators, why is it that there is very little information on the biology and the ecology of that particular area? They didn't have much answers. They said, well, we hired the best consultants and this is the best report that we got. Over a few months, I had the opportunity to visit the country where these consultants were located. And I went to their office and I talked to them. And I was surprised to note that they don't have a biologist. They don't have an ecologist. They don't have a taxonomist. They don't have any of these specialized fields on which basis they would use and write biodiversity information. So the data was not being reflective of the impact assessment they were submitting to me. So I'm putting this to the audience, to this esteemed audience that I have right now, that this is a subject that you need to relook. we cannot look at chemical analysis as the ultimate impact assessment, or look at physical analysis for impact assessment. We need to look at more on the ecological part of it and the biology part of the area in which these projects are located. I'm taking this example for those of us in India and in Maharashtra in particular. You may have read in the news about this particular lake located somewhere in Maharashtra that turned pink. I want us to recall that the news came because the color was pink and it was a large water body. It has become pink for a few days and then it will change its form. But before it became pink, we need to understand that an ecological process of succession has actually taken place in this lake. All right, so we've had things like some form of vegetation dominating over a period of time, like algae, blue-green algae. Then you would have had an algal boom. The algal boom would have deprived the water body of dissolved oxygen, and the algal boom would have died and become nutrients. And the nutrients would have formed some kind of energy level for some kind of diatoms. And therefore, the prominence of some certain diatoms that had this color made this lake look pink. So the concept of succession is something that one needs to understand. And then you can understand why this lake became pink. I have another example, which some of you may have heard. We've had flamingo birds coming to Bombay, especially on the Palm Beach Road. And the numbers that arrived here, you will not believe it. There are like 100,000 or 50 to 70,000 flamingos. Understand that the flamingo is not a small bird. It's actually a bird that is about 4 feet tall. Okay, 1.2 to 1.4 meters tall. It weighs about 2 to 4 kilos. And it has traveled a long distance and it has found this particular water body. The water body is probably 18 inches to 2 feet in depth, spread over 10 hectares or 20 hectares of land. And it's just a water body which normal people in the 365 days would not have noticed this to be of any significance, except for the 4 days that the flamingos arrive at this spot. Now, the 4 days changes this particular shallow water into this beautiful pink-looking avian environment. Understand the size of this bird and assume how much of food they need. They have actually stopped in this water body as a wasteland to eat. So the amount of food that they have here, which is basically composed of blue-green algae, red algae, diatoms, larvae, the adult forms of small insects, crustacea, mollusks, and small fish. So much of biomass, so much of food was available for these birds to come here. And therefore, they have come here en route to the migration that they were going through. I put this across to you is because out of the 365 days, these birds are only available for 4 or 5 days. And therefore, if one doesn't see the seasonality of the ecosystem, we wouldn't appreciate that this is a very important biome or a very important habitat for this particular type of birds en route in migration. To bring it further, I want us to recall basic concepts of ecology. Important, we are all familiar with things like food web, food chain, trophic levels, energy pyramid, biomass pyramid, pyramid of numbers, species diversity, relationships, whether they are symbiotic or parasitic. All these concepts are easy to understand, but in the point of project management or while developing a project, we need to see and understand exactly what is the food web, what is the food chain, what are the trophic levels available, what are the communities, what is the population, how does the population dynamics or ecology work. It's all this that is more intricate to that ecology. Understand the ecosystems is nothing but a whole lot of niches and habitats that we have. So I want us to recall that these concepts are not textbook concepts, but these are concepts that actually happen in real environments. And we need, as environmentalists or life science teachers, students and professionals, to understand that it's essential to relate it to impact assessment. I'm coming back with this background to the Niger Delta. This is the place where I actually did my master's thesis. You can understand that this is a very huge delta. It is one of the most unique deltas in the world. It's a very stationary and static delta. In other words, it doesn't move. And because it is stationary and static, over a period of time, there is a lot of anaerobic conditions. And so what changes in the sediments and what changes in this saline environment is so unique. To put it in perspective, the Niger Delta is about 70,000 square kilometers. It's on the equator, so it is more of an equatorial forest. I want you to note that some of the mangroves in some of these regions of the Niger Delta grow as tall as 60 feet. In some places, maybe reaching 80 feet. I don't know how many people have noticed or heard of mangroves growing this tall. Most of the mangroves I have seen on the Indian coastline are shrubs, relatively 10 feet, 15 feet in height. They don't seem to have depth. From the coastline, they would stretch maybe half a kilometer or less than that. We have fringes of mangroves, but this is an area in the Niger Delta where for at least 100 kilometers or more, the mangroves actually have an influence. From what we know, this delta of Nigeria is very rich in species diversity. We have over 4,600 species of plants, 206 endemic, out of which we have 484 species in 112 families, which are threatened to be extinct. To make this delta more illustrative, we have a zonation system here. The outer fringe of the delta is mostly sandbars and the ocean on one side. And then as we get towards the deltaic area upwards, we see that we have different types of vegetation. We have the mangrove.
We can't see the screen, sir.
You cannot see the screen?
Yes.
All right. Can anybody see the screen or is just one person?
It's just tiny.
Sir, we can see.
It's a tiny portion. It needs to be made to be bigger. Let it feel the
Okay. So,
Hello.
yeah, can you see now?
The screen is visible, sir. All the participants, please requested to pin Avinash Dulip, sir. He is presenting now. So you will see the another user, Avinash Dulip, presenting. You need to pin that user only. Is it okay?
All right. Is
Yes,
that better
it's
now?
visible
You have
now.
the screen?
Okay, shall we start, sir?
Yeah, I'm okay. Is everyone okay? Can you see the screen? All right.
Okay.
So the mangroves, you see, is a very vast area in the Niger Delta. It has very unique conditions of soil. Over a period of the seasons, the soil chemistry changes, which I'll be getting into in detail. Understand that the mangroves is basically diannual tidal regimes. That means you have 2 half tides and 2 full tides that come in every day. We also have the lunar cycle. So we have nip tides and spring tides. All right. And this flows into the river bodies across all the rivulets that you see, including the main river, which is the Niger River. And because of this diannual flow of tides, the estuarine ecology becomes very dynamic. It changes every 12 hours, every 6 hours, because the tidal flats are flooded in some point and then they get exposed. So you get anaerobic to aerobic systems all the time. I put this picture in your front just to give you an idea that we are located in a rainforest and there's a very rich mangrove forest here. In this Niger Delta, I've also superimposed a rough map of oil and gas oil fields. We have over 1,000 oil fields here. We have 100 process plants. We have 1,500 kilometers of major pipelines and over 45,000 kilometers of flow lines. I want you to understand that in the 70,000 square kilometers of the Niger Delta, you have such large amounts of infrastructure. We are not talking about, jetties. We are not talking about drill sites. We are not talking about roads. These are all other infrastructures that are there. We're not talking about offshore facilities like SBM, loading, unloading jetties. So there's a lot of heavy oil and gas activity in this area. I have in the next 4 or 5 slides to give you an idea. There was some data that was available where the Niger Delta was studied. We see that this is a pH data points of different parts of the delta. I've written some in very high letters. So you have some pH that is 5.4, 5.2, 4.1 in some places, 7.2 on the coastline, 7.6 towards the delta. This pH indicates that every unique ecosystem or riverine system is very specific to itself, in as much as it is within the deltaic zone. Okay, so the pH is very important, and you can understand that they vary a lot. Now, the bottom side of the slide is where the Atlantic Ocean is, and the top side of the slide is where the land mass is. All right, just to give you an idea that the rivers flow from top to down. All right, so likewise, the salinity would also change from top to down. The next slide gives us some data about total extractive amount. This data is very old, but just to give you an idea that within the large infrastructure of oil and gas, there is this data available that indicates the total extractable hydrocarbons in water, which is the top figure, and in the sediments, which is the bottom figure. All right, and it ranges from 17 ppm on the top, on the surface in the water, and in some sediments, it's as high as 878 ppm. In some areas on the left-hand side of the screen, where you have a lot of oil and gas activity, you have 1,000 ppm. This is old data. I'm sure the data is probably better or worse than what we have here. We have pictures of dissolved oxygen. Now, dissolved oxygen gives us the health of the basic riverine systems. So we have a place that is 7.9, just to give you that it's so high. Normally in water bodies, we have 3 to 4 milligrams per liter. It's supposed to be a very healthy water body. Now you have 7.9. In some cases, just 2.5. All right, and 2.5 indicates that the dissolved oxygen is a little bit low. And more often than not, the 2.5 may not be near oil and gas field. It could be its own very unique ecosystem because of the type of vegetation that grows there. It could be leaching out some form of acidic leaches. So the pH could be as low as that. We also have TDS and conductivity. So the figures on the top talk about total dissolved solids, and conductivity is on the below. It's basically related to salinity. You will see that as we go from bottom to top, the salinity decreases because the riverine system or the estuarine system is very unique to this situation. Interestingly, this forms zonation. So you have different salinity zones across this particular area. So this delta, you'll notice that the salinity changes from open ocean. Yeah. Anything else?
Sir, sorry to interrupt you. Sir, screen is not visible. Presentation, we can't see the presentation.
Okay. I'll try to get it
Hello?
better.
Hello?
Yeah. Can you see this?
Hello?
Yes? I can hear you.
The screen
Yes,
is
sir,
visible,
it's
sir.
absolutely visible. Yes, sir, visible.
Okay.
Thank you.
Please carry on, sir.
So the 3-4 slides I gave before this were talking about pH, dissolved oxygen, conductivity, total extractable hydrocarbons across the delta. To give you an idea that from the open oceans all the way up to the riverine, the information changes. Now we come back to the actual forest, which is the mangrove forest. And I need you to understand that This forest is entirely rich. The photograph I have on the right side, this is a mangrove tree that is as tall as 40 feet. And if you walk into this forest, you'll find it so dense. And actually, it's pretty scary to be in this part of the forest. We also see on the left side, there's a mangrove with a lot of oysters on the roots, and there are barnacles also on the roots. Why I'm sharing this is to tell you that we have very exclusive niche in the mangroves. We have habitats that are very unique to the substrate along the channel of the estuary, along the intertidal flat towards the supralittoral or towards the land. So each of these habitats are very unique. They have different types of vegetation, different types of fauna and flora, and the soil types are also entirely different. So I'm going to illustrate in the next few slides how this is. The sediment types, mostly in the high and mid intertidal, are dominated by Avicennia, which is this type of mangroves, white mangroves as we call them. And the white mangroves are mostly on the middle and the high littoral zone. You have the red mangroves, which are pop roots, basically towards the river channel and the lower littoral zone. So if you look at this particular terminologies, but one is submerged in water all the time, and then every nip tide and spring tide, the water goes to the fringes, and this is very remarkable. The high tide on a nip tide reaches a certain height in the littoral zone. On the spring, the tide also reaches to a much higher zone, which means these roots get exposed, which are on the far littoral, once every 28 days when there is a spring tide, and some of it would get exposed on the nip tide, which is in the mid littoral, every 14 days. Okay, so the plant and animals that survive here have to survive with this phenomena of high tide and low tide during spring and nip tides. The soil type that we have in the red mangrove area is mostly of a mat, which is called a thick peaty texture. It is called chikoko, and the spongy chikoko absorbs considerable amount of water. For those of us who walk in this particular substrate will understand that it retains a lot of water, and the weight could be as much as 6 times of water volume it can retain. And they use chikoko basically for aquaculture bunds. If those of you who have built aquaculture bunds will understand how that is being used. In the Avicennia, which is the high littoral, the substrate type is mostly sandy and are not very fibrous in nature. So that is how the substrate changes along the fringe. I want us to look at these two drawings here. The right hand shows a low tide, and the top figure shows a high tide. I want you to understand that when the tide starts flowing in and you start observing, it takes a long time for this channel to fill up. After the channel is filled up, it goes on and splashes onto the tidal flat. Okay, so the expanse of land suddenly becomes huge, and it takes a longer time to fill the tidal flat. Okay, so the time frame between low tide to high tide is very significant to the types of plants and animals that grow in this particular area. Understand that the tidal flow that comes in has its own time constraints during nip tide, where the soil is exposed to air more, so it is more aerobic activity. Whereas in the spring tide, the water is exposed, the water is more on the land, and therefore it becomes an anaerobic situation for a much longer period of time. So this is a particular specific area that I had during my course curricular I have studied, which is called the New Calabar Bonny River. And this is an area that we have extensively studied across our estuarine ecology course. The reason I'm putting this across is when we study life sciences, more often than not, ecology seems to be a subset. It's a paper only. It's not a full-fledged course. And therefore, the information we get is mostly for for that one semester. But it's so critical to understanding it. As we go along, you will begin to appreciate how much effort goes into understanding this particular ecosystem. I have marked out an area where I had actually studied. These are slightly old photographs, but you can see the Avicennia africana with the Pneumatophores. You can see that you have sandy substrate and you have muddy substrate. You also have the pop roots of Rhizophora, and you also get some of the tidal flats with algae. Some of them don't have algae. All right, and some tidal flats are just with silt. There is no vegetation at all. So all these types of substrates I had studied, and I have tried to document it in terms of biogeochemistry of those particular substrate with reference to the major types of vegetation on them. This particular iron structure you see here is actually a corer which I had designed to take cores of the substrate up to 10 inches, okay, of the substrate. Some of the parameters I studied were basically particle fractionation through sheaves, conductivity, total organic content, DO, BOD, sampling of microfauna, and identification of species. Each one is a very big challenge. To come back and give you some of these particular examples this is Avicennia soil, this is Rhizophora soil on a nip tide. It's an illustration I'm giving here. The water was covered from this time to this time, and this time to this time. Okay? And if you look at this particular, where my arrow is, this is the lowest tide. Okay? And this is the highest tide here. Okay? So if you look at the temperature in the soil, you'll see that at the lowest tide, the temperature was low, and it became very high just before the water cover or the tidal water came up. Likewise, redox potential, which is a measurement to understand whether it's oxygenated or deoxygenated, you will see that just when the water is covering the soil, the redox potential goes negative or goes below. The pH also reacts. The three lines actually indicate the top soil, the middle soil, and the bottom soil. So the top soil is actually the soil at the surface. The bottom soil is about 12 inches, okay, below the surface, and the bottom is about 20 inches below the surface. Okay, so these are substrate soils that I'm talking about. In the Rhizophora spring tide, all right, you'll see that the water cover is of a longer duration, unlike in the nip tide. Okay, so I have only temperature here. All right, so you can see that when the tidal flood gets exposed, the temperature increases of the soil. There don't seem to be much difference to the bottom layer of the soil. This is again Rhizophora spring tide, just the redox potential and the pH as they vary as we go along. We also have, interestingly, a seasonal variation. So I have from June all the way to December here. I also have a nip tide and a spring tide, a nip tide and a spring tide below. All right. And then I have a pH reading here. I also have redox potential, and I have BOD, or dissolved oxygen, in BOD on one side. You will notice that in the month of August, we have rains in this particular area. And because there is fresh water influx of flood water, a lot of these parameters change. So there's a rush of fresh water over the saline water. And the sediments and the water actually change its properties. Okay? Fortunately for me, in the month of October, we had some kind of a petroleum spill. And the effect of this petroleum spill was noticed for about 3 weeks before it came back to normalcy. And you can see that by different values that I have here. We also noticed that in the months of November and October onwards, you will see that this is mostly a season that is cold or winter. That is very different from the dry season that we have at the beginning of the year. This is about a silt zone without any vegetation and how the parameters have changed along the seasons. They react differently because they don't have vegetation at all, and they are very fluid in nature. This is a mid-intertidal. Mid-intertidal, also you can see there's a little bit of difference in how the floodwaters react to different parameters. So there's a comparison between high tide, mid tide, and low tide to understand how these parameters have changed. I've done pH, redox potential, and BOD profiling for different depths, okay, across different months. So this is June, July, August, September, October, November, December, and for all of them. I've only put it here as a representation of the amount of work that went into it. And based on the kind of analysis and observations one has had, there's a general trend that we get. I have noticed that the total organic content of the mid, high, and silty intertidal zones are about 45%, 24%, and 13% respectively, and includes a lot of roots and fibrous peat material in the substrata. In the silt area, there are certain clay components that have the ability to swell on hydration. We see that the pH is generally acidic, except during the month of September when it is alkaline. The redox potential shows that the substrate was very reduced from 10 to 450. In the rainy season, the substrates are in sulfite system, which changes in dry season to organic acid synthesis. This is reflective by the change in color of the substrate. In rainy season, the substrate color is black, and it's dry. In dry season, it is brown. We'll get a little bit more on the different colors. The BOD is generally in the range of 3 to 4 milligrams per liter. And because I had an oil spill in the middle, I was able to understand how the pH fell down from 5.2 to 5.4 and how the redox potential changed from 490 and the BOD became very high from 5 to 7. And also reflected that there was a change in the way the oil spill had recovered in those substrate. So in summary of having gone through this work, there are two things I want us to understand. On the right side, I've seen thin brown green layer and a blackish layer. This is, to an observant eye, you will notice that within the first half or 1 inch of the topsoil, we see these changes of color. These changes are basically reflective of the type of algae that grows on top of them. So on the surface, we have a thin brown layer followed by another thin green color, probably due to blue-green algae, plus bacteria growing like. This is followed by an even wider range of blackish-gray mud. As we go deeper into the substrate, like 14 inches or 20 inches, we'll see that all these parameters that we have changed. You can see the way there's a redox discontinuity or reduced state, and therefore this color has changed from yellow, gray to black. And that's what really happens. The critical environmental factors that most likely control the rate of organic degradation in estuarine sediments are the presence of an active microbial flora capable of degrading the individual organic substrate present in the sediment, the oxygen regime within the sediment and in the immediate overlying waters, the availability of nutrients other than oxygen, bioturbulation and the level of dissolved oxygen determine the type of microbial population which exist in the sediments that determine organic degradation. This is very critical when you look at the redox potential and the different colors that we have in the substrate. As dissolved oxygen concentration is lower, there is an increased tendency of incomplete oxidation of organic substrate, and in the complete absence of oxygen, it might be producing methane. The lowering of redox potential within the sediments with increasing oxygen depletion results in a stepwise microbially mediated process of denitrification, sulfide reduction, and methane formation. So this is what really happens in the mangroves, and it's very critical. Now we need to look at these conditions under which the flora and the fauna exist. You need to understand few things. The exposure of aerobic to anaerobic changes dramatically over a period of 1 day, over a period of every lunar cycle, and therefore the flora and fauna have to adapt to those changes. And that is how complex the estuarine system is. The biological components that were studied during that time were basically based on the substrate type. So in the high intertidal zone, we had the sandy substrate, we had the muddy substrate. The flora and the fauna are entirely different. In the mid-tidal, we had the soft mud under the pop roots. We had algal covered substrates and non-algal covered substrates. In the silt zone, we had a flocculent silt. So all these substrates were analyzed for the type of biological components that they have. And the main taxon that we found were mostly mollusks, which were gastrocopod bivalves. We had annelida, which are polychaetes, and arthropods, like insects and crustacea. I have a table here that may not be very clear, but basically this gives me the substrate type: sandy, muddy, soft mud, algal cover, non-algal cover, and silt. And these are the different classes. So I have gastropod, bivalves, polychaetes, insecta, and crustacea. These are different types of species that we have identified, and the abundance or not abundant are marked by the number of stars that you see for each substrate. This gives a relative idea of the abundance of this particular type of flora and fauna that we have here. We also find that we have a crab community that is well developed in the high intertidal zone, in the mid-tidal zone, and in the silt area. So in the silt area, we only find it at the banks, which is basically uka, tangari, which is a crab that actually goes both on the land near water bodies and also towards the estuaries. But in the mid and high tidal, we find that we have different types of crabs there. Interestingly, the breathing roots or the pneumatophores of the Avicennia, which are spread in the mid-littoral to high littoral, either on muddy or sandy substrates, have a very distinct vertical zonation of flora and fauna. It takes a lot of effort to really understand what goes on and around these breathing roots. Basically, the breathing roots are used for the plant Avicennia to breathe and be healthy. By virtue of high tide and low tide, a lot of estuarine organisms have adapted to find a way of hatching, laying their eggs or their small forms, so that they have a very safe haven. So take the eggs of some crustacea, for example. They swim here on the spring tide. They lay their eggs on this moss that is made up of Rhizocolium, Brochunium, and Colagosa species of algae. You have tunics. You also have some kind of Archachadia, which are mites that also lay their eggs. You also have some polychaetes. Now, after laying their eggs, they remain in this place without water for the next cycle of springtide, which means they have that much of 28 days to hatch. So when the next springtide comes, the hatchlings are ready now to swim with the water. And that's how the mangroves actually become a breathing and a nursery and a hatching ground for a lot of marine species. At the bottom of this particular route, we also see that we have a lot of gastropods and sea ammonites and mats of rhizocolium. Okay? And if you keep looking around this particular area, you will see that this is almost very apparent along the coastline, that wherever you have Avicennia roots, you have this kind of vertical distribution of selected algae and invertebrates. It's very unique to this particular system. Understand that it's the type of flora and fauna that stay here are dependent on the neap tide and the high tide, which actually influence their hatching biology or their cycle, and also when they will be released back into the area, which is the water that they live in, okay, over a period of time. Likewise, in a pop root, the red mangroves, there is also a vertical zonation. The red mangroves are a little bit different. Notice that this type of pop roots have probably more fauna than those on Avicennia roots, because the water is more in contact with these roots, because they are located more on the channel of the creek. So the water, they're almost submerged more in water than the Avicennia roots. And therefore, a lot of animals of flora come here to lay their eggs, to hatch, and breed, basically. So these pop roots form a microsome for these particular types of animals. So you have a lot of oysters, barnacles, isopods, microcrustaceans, coral strains, eggs and larvae of fauna. You have some crabs, you have mollusks, you have gastropods. I need to indicate that in some areas of the Niger Delta, we have seen corals that also get attached. And it's amazing to notice that in blackish water, we have such brilliant looking corals. I'm not sure what is the situation today, but I remember when we used to study the Delta a long time ago, we had significant amount of corals also attaching to the pop roots. So to put this in perspective, I want us to recall the estuary is very dynamic. It varies in salinity from 35 ppt to freshwater. It also has horizontal zonation from the channel of the water all the way to the supralittoral part of the high tide. And we also have vertical zonation, very unique. You've also seen the Rhizophora forest and the biochemistry of the sediments. We've also seen the pop roots ecosystem, what they have, and the biochemistry of the pop roots. We've also seen the regime of how much water covers for how long along the littoral flat that we have. So all this is very important when we do development of oil and gas, rather than saying things like the impact is possibly loss of biodiversity. The word loss of biodiversity is too broad, it's too generic. We need to be specific in terms of and the habitat that we're looking at. What if one particular habitat does not exist anymore? What will happen to the ecosystem is what we need to be looking at. We don't look at greenhouse gas. It's a very global thing. We need to look at very specific areas that the mangrove actually absorbs a lot of methane. Okay, and therefore absorbs a lot of carbon dioxide, and therefore it's a very good sink for a lot of carbon dioxide. Okay, so we need to be very specific in what we're looking at in this particular area. With this, I actually come to the conclusion on this first part of my talk, which is basically on estuarine ecology. I have not spent a lot of time on oil and gas activities, but yes, oil and gas activities are spread across the delta in their various facets. I've tried to emphasize the importance of biology and ecology when we look at impact assessments, and particularly to the students who are studying life sciences, to the faculty. It's a challenge. I'm challenging you that you need to focus the curricular to reflect actual fundamentals and principles of ecology, so that when the students graduate out, they understand the importance, rather than being very generic in their in their outlook. I also challenge the consultants who prepare the environment impact assessments that you don't look at biology as loss of biology or loss of biodiversity. You need to be specific. You don't list flora and fauna. You need to really understand what is the food web, what is the food chain, what is the energy levels that we have, what are the habitats, what is the unique habitat. If you remember in the beginning, I showed you a slide of the flamingos. Now the flamingos only spend 4 days in that shallow water. All right, the pink coloration of the lake. Okay, it's just a phenomena for a week. So we need to understand ecology from that point of view for every site that we study. Okay, so with this, I'm going to go to the next topic that I have. And I would prefer that we have questions probably at the end of this presentation. Okay, so I'm going to the next part of it, the significance of ecology and biology in oil and gas projects, the Indian Ocean, in particular the Arabian Sea, the Western Indian Ocean. The Western Indian Ocean is a very vast ocean, as you will see. It started operating oil and gas activities in the mid-1970s. It produces about 20 million tons of crude oil annually and about 20 billion cubic meters of natural gas annually. To give you an idea of how large the activities are, at any point in time, we have almost 6,000 to 7,000 personnel staying offshore. And in some cases, it goes up to 10,000 people staying in the high sea every day. To give you an idea that we have a mini township offshore, and it's a very large operation that we have here. And they are spread over a large geography, very, very large geography. The ocean is not as simple as a river or an estuary that has a finite measurement. An offshore is a very large water body. And I've just given this map here to show that there is a depth profiling, and the ocean is extremely large, and therefore studying the ocean with reference to oil and gas is a very challenging job. What is it that you're looking at? How is it that you want to look at those parameters that you want to see are critical? The Indian Ocean currents are large. All right, you have the northeast monsoon, you have the equatorial counter currents, and you have all sorts of currents that change with seasons. And they flow across this large mass of water body. Now, the currents, when they flow, I want you to understand that if it's pre-monsoon or during the monsoon, the currents will behave in a certain direction. In others, they will behave in a different direction. And this will determine what happens to the phytoplankton, the zooplanktons, the crustacea, the fish, the mammals, the reptiles, that exist in the ocean. The circulation patterns in the Arabian Sea is very critical. This has been done by a lot of oceanographers. There's a lot of study. This is not my study. I'm only reflecting what published data is at the moment. We'll see that we have WICC, which is Western Indian Coastal Current. In the winter month, it goes from the south to the north. And in the summer month, it goes from the north to the south. All right, so you see that there's a reversal of this particular current. We also see that we have a Lakshadweep high in the winter and a Lakshadweep low in the summer. We also have a north equatorial current that flows from east to west. And we also have Indian monsoon current that flows from west to east. Now, these patterns of circulation actually makes the ocean churn around a lot. And churning around just not the surface, but also the sea beneath, underneath in depths. So there's a phenomenon called upwelling that occurs. Now, upwelling is basically a process where the surface water goes underneath and the down water that is at a depth comes up. This happens during the monsoons, and this brings a lot of nutrients along with it because it churns the water body. And with these nutrients, we notice that after the monsoons, we find that the fish catch is normally high. So from October to March, you get a better fish catch than during the monsoon season. This brings in a lot of amounts of nutrients into the upper ocean, which enhances primary productivity, and this contributes to the fisheries along the west coast of India. The fish catch is highest between October to March. From an oceanographic point of view, the Arabian Sea is divided into two regions, 15 degrees, south of 15 degrees and north of 15 degrees. We notice that the fish caught in the south of 15 degrees is of a different composition than the fish caught in the north of the 15 degrees. We find that in the south of this line, we have phytonivorous fishes are dominant. That means plankton-eating fishes are more below the 15 degree. And in the north of the 15 degrees, we have both the carnivorous are abundant and the planktonivorous. Along with the circulation, we need to understand that there are a lot of different forms of animals in the ocean. And those who stay offshore will see some of them pass by. Either they're migrating or they've come around the platform for food or some stuff like that. You get a lot of birds that fly. And it's interesting to see the journeys the sharks make, the whales make, the turtles make, the dolphins, even snakes, reptiles, and the birds. It's phenomenal. So in this dynamic Arabian Sea, how does one understand whether the environment in which we are working, the oil and gas activities, is managed well or not? So we've used environmental monitoring as a tool in understanding this. And we find that this is a very variable tool. But in this vast open water, how do you define monitoring? If it was on land, you could have some kind of a system to understand it because you can stand on land. In the open waters, it's almost impossible to know. So there are some methods that have been designed. Okay, I'll just go through this quickly. Normally, you have, there's a small schematic for especially the students who don't understand oil and gas activities. We have drilling activities, we have operating activities, we have transportation, we have production. So all these are operations. Each of these operations will have some source of emissions. They'll create some noise, there'll be sewage generation, there'll be solid waste, there'll be effluent, there might be possibilities of oil spill. So based on the type of activity or facility, you matrix out whether any of these elements will be affected or not. And invariably, you can find out whether they're affected or not affected. Then you look at, for these elements, what is the quality that might be affected? For example, if the oil spill occurs, it will change the water quality. All right. If the effluent is discharged that is not treated, it will affect the water quality. So you have a matrix of these elements against some parameters like air quality, water quality, sediments, plankton, benthic biota. Okay. Now, interestingly, the regulation would talk about mostly the air quality and water quality. It may not have much to talk about sediments or planktons or the biological systems or ecological parameters. Okay? And that is where the challenge is. Now, if, for example, the environmental impacts of these operations are there, normally the targets of the change in the quality of air or water or sediments or biota would be, it will affect either the hydrology, that means the water body itself, or the nutrient levels, depending on what is being discharged from the platforms and the rigs, the hydrocarbon content. We use a lot of chemicals offshore, so there will be metals and heavy metals. Will it affect the biology? So this is a matrix that shows an environment impact and monitoring matrix to give you an idea of what we need to look at. So the impact assessments are basically based on this matrix. And then looking at the severity and the consequences, they would rate it. But today I'm trying to restrict my talk to mostly the biological and ecological parameters and understand that they are as important as the chemical analysis that we do. So the parameters that we study are basically for understanding the impact of oil field operations on marine environments are recorded in things like hydrography, which is basically parameters related to circulation, stability of water, column, the physical characteristics of marine environment, such as salinity, temperature, turbidity, dissolved oxygen, pH, suspended solids. In the nutrients, we see that these elements are essential for plant growth, such as nitrogen, phosphorus, and silica. The concentration of these parameters generally govern the primary productivity of the region, and they are season dependent because of upwelling and downwelling of the oceans. A lot of churning of water and ocean bodies take place. We also may have some pollutants, concentration of heavy metals and petroleum hydrocarbons in water, sediment, and in the tissues of the fish and other living organisms, including phytoplankton and zooplankton. We also do studies of biology. So we study biota in water and sediments, population abundance and distribution of the plants and animals in the water and sediments. We measure chlorophyll A, phytoplankton, benthic biomass, and stuff like that. To give you an idea of how far the operations are, they are about 80 to 100 kilometers in the offshore at a water depth of about 80 meters. And they expand a few hundred kilometers north and south, lengthwise and breadthwise, maybe 40 or 60 kilometers, to give you an idea that the geography we're talking about is large. And therefore, well, this is just a Google map that this is Bombay and these are where the oil fields are. So they are very well spread out, not very near to each other. So for us to put everything in perspective, what we do is in the monitoring program, we design a radial type of sampling. So the epicenter, the central point is actually the platform, the rig, the facility of oil and gas. And then we have a 500-meter radius around it, a 1 kilometer and a 2 kilometer. We also try to divide it into north, south, east, and west, trying to avoid the pipelines that are flowing below the subsurface. So looking at the subsurface, we need to understand that there's a lot of underground infrastructure that is there. So while taking samples, we have to avoid those places so we don't damage them. So the first thing is we do radial sampling, 500, 1 kilometer, 2 kilometers in this format for almost all the rigs and platforms. And across the facilities, we have a grid type of sampling, wherein we have control and reference stations far away from the field, the oil fields, which become reference stations. So the grid also gives us an idea of what is happening, and we are able to interpret the information and data based on radial sampling and grid sampling with reference to the actual parameters that we measure in the initial phases, which is the nutrients, the pollutants, the biota analysis. We also measure them across different depths. So we have surface samples, we have middle samples, and bottom samples. We also have water samples and grab samplers. So water samplers are for water, and grab samplers are basically for benthos. We also follow the season because you'll notice that in the season, the parameters change. So we have a pre-monsoon, which is March to April, and a post-monsoon, which is from December to January. We don't do sampling during the monsoon because it's very rough sea. So we have mostly the sampling between pre-monsoon and post-monsoon. On the research vessel, we need to have inbuilt instrumentation. So we have current temperature depth profiler, Neskin samplers, Van Ven grab samplers, you have snappers, you have platform nets, you have a GPS, of course, you have a computerized weather monitoring station, and many other facilities, including analysis. Some schematics, some pictures to show what we do, and equipment that we use. So we have a CTD profiler, we have a current flow meter, we have Neskin samplers for water, we have sediment sampler for grab samples. Now, a sediment sampler has to travel 80 meters in the water to hit the benthos and have an intact benthos sample to come all the way up of 80 kilometers of water column. And that's very important. And it's not an easy task to take those samples. Some of the methods and instruments that we use in the study, these are very standard. I've only put them here for illustration. So for salinity, you can use a salinometer or do a titration method. pH, we have a direct pH meter. Turbidity, we have a meter. Dissolved oxygen, we have a titration method. For nitrates, nutrients, in general, we use spectronic method. For chlorophyll, we use a spectronic. Petroleum hydrocarbons, we use a UV fluorescent spectrophotometer. For heavy metals, we use a pre-concentration. And for heavy metals, we digest them in some acid and preserve them for analysis in the ICP spectrophotometer. So on board, you have a lot of equipment. And I want you to understand the ship that you have this equipment is rolling and pitching with the waves, all right? Which means there is a lot of, the boat is not stable, all right? So while you're sampling, while you're doing analysis, everything is not an easy task. But however, you need to carry out this function. And most of the equipments are tied, so they don't float around, but they're fixed to a particular structure. These are some of my colleagues. There's Dr. Sen Gupta, one of the greatest oceanographers that we've had. Neskin samplers, plankton samplers, benthos samples. Benthos samples are not an easy task because after the benthos comes up, you have to put it through sieves to pick up all the different flora that is associated in it and preserve it in formalin. I've given this as a data of pre-monsoon from 1995 to 2001. I have parameters on one side, I have the years on the other side, and I have reference stations here. So that gives some idea of what, I'm not here to discuss the results, but just to give you an idea of how offshore monitoring is a good tool for environment management with reference to oil and gas activities. We also have post-monsoon data, which is very unique compared to the pre-monsoon. And generally haven't done this study over a period of number of years. We find that the offshore operations, because the offshore operations are such that we don't allow normally anything to leak into the water or fall into the water. Everything is a closed loop system or cycle. So nothing really goes off. We find that there is very little or no effect on the offshore operations on the general hydrography of the water column. There is availability of essential nutrients for plant growth and biological productivity. Most pollutants were well within background levels because we had reference stations, and most metals were observed to be very, very low. So I have finished giving you my two presentations, and I'm actually very open to any questions and answers. So I leave it to the floor, to the convener to decide how we can go about that. Thank you.
Thank you so much, sir, for this very informative session.
Thank you.
Sir, we have some questions from the participants. Can we have those questions, please?
Yes.
So the question is, can you give some examples of mangrove plant species which can be easily grown in desert and which can be cultivated or grown around the water sources of Rajasthan, especially Western Rajasthan?
Okay. Normally, mangroves are are hyaline, that means salt growing plants. All right, and they grow in saline waters. I am not aware of any species that will grow in the desert. I'm not sure if there's anyone among the audience that has any idea. But from my side, I don't see any species that I'm aware of that can grow in the desert.
Thank you, sir. And the next question is mangrove preferable in pharmaceutical manufacturing?
That is an area I'm not very conversant with. I have read that some components of mangroves are being used to make dyes and some pigments, but I'm not very sure of its other properties. I've also heard that in certain mangrove forests there are honey bees that grow honey, and that's a very unique type of honey that grows in those areas. That's all I can say. I am not aware of any pharmaceutical use right now.
Thank you, sir. I thank you on the behalf of Dapoli Urban Bank Senior Science College. The feedback link for the participants will be shared on WhatsApp group. Certificates will be mailed to you in 2 to 5 working days. And participants are requested to fill the feedback forms in 1 hour strictly. We oftenly organize such webinars. We have our YouTube channel, which is webinar DUBSSC. So please, if you like this activity, subscribe to our channel and like our videos. Thank you.